I want to point out, so we do a lot of technical things here, so just show of hands, how many of you are familiar with developing analysis in SAS? R. Raise your hand if you're an R person. SQL. We're doing some web app stuff. Who's working on JavaScript? Who's an Angular person or a Knockout person? Okay, who's writing MATLAB? Stata. Okay, those graphs that George showed, he programmed himself in APL. Show of hands, who's an APL person? Oh, we got one in the back. We got one in the back. All right, two. We got two. Did I hear three? No, okay. Well, you are not alone. You're in a community that embraces all thoughts and perspectives and ideas, no matter what they might be. All right, so I want to get started on this journey that we're going to try to walk you through. So George told you that we're really trying to sharpen the knife in terms of what the focus of this community is, and it's about generating reliable evidence. And we've used in platitudes these terms: clinical characterization, population level effect estimation, and patient level prediction. But we really want to dive in. We've heard from the community, people say like, yeah, we get it, but what does that really mean? And so what we really want to do this morning is provide you a very deep dive in what we think is a perspective of what it could mean, what it has meant to us, and posit a future direction for all of us to follow in terms of where it could be for us as a community. And so I put this up every time. Odyssey, you look it up in a dictionary. It's defined as a long journey full of adventures and a series of experiences that give knowledge or understanding to someone. And so I think that this is very pertinent to our community. Many of you know that I keep pimping it. You all should read this if you haven't in high school. To read about Odysseus' travels. It's a pretty circuitous route that he took from the war in Troy back home to Ithaca. But there was quite a lot of discovery through that process. We've been talking about Odyssey's, the community's journey. What I want to pivot to a little bit today is talk about the journey of an individual patient. So I want you for a second to think about yourself, maybe a loved one, a friend and family, somebody who might be experiencing illness and is going through the health system. And what we want to talk about is that journey, and both what does that experience look like and how we can learn and both contribute to that patient's welfare as well as learn from their experience so that we can contribute to other patients like that. So to do that, I want to provide a caricature of what a patient journey might look like. So what I'm going to show you here is just one individual patient as a simple little cartoon. On the x-axis I've got time. On the y-axis I'm going to show you various domains that we might have information about a given patient. So we might have information about conditions, drugs, procedures, measurements, other types of data that are captured in clinical systems. But fundamentally to a patient is just a person living their lives, having fun with friends and family, and at some point in time they develop an illness. They have a disease. That disease might be recorded as a condition in some clinical system. But ultimately, the patient doesn't care about the data going into a system. They care about getting better. So they seek they seek an encounter with a healthcare provider who's looking to help remedy that disease. That remedy might involve treatment of some sort of an intervention, whether that be a drug or a procedure, some intervention where the doctor is going to do the best that they can using the science that George outlined to think about what's the right treatment to treat that disease. And at that point in time, the patient's life is going to go on, and all we're generally concerned about is what is the outcome going to be for that patient? Are they going to get better? Are they going to experience some sort of other events? Now this is the journey of a patient manifest in data. There might be data in a clinical system about the disease. There might be data in a clinical system about the treatment. There might be data in the clinical system about the outcome. But of course, there's also going to be other data within this patient journey that's captured in various clinical systems. So there might be other diseases that patient has experienced in the past. There might be other treatments that they've taken for other conditions. They might have administered various procedures. They might have had measurements. A patient's journey manifest as data can kind of look like one of these cartoons of a collection of data time-stamped over time that we're going to try to learn from. Now, realistically, what we want to think about is that a treatment isn't just a data point. A treatment's a decision. a decision that's being made by the patient in collaboration with the provider to think about what do we know up to this point so that we can make a decision about what's going to happen in the future. So we can think about, when we talk about data, we can think about analysis being performed using baseline time, the data that's captured before a decision point, and we can think about the follow-up being when are we going to be looking for a given outcome. So this is how we can structure data, but ultimately to a patient, they've got an illness, they've got a decision to make a treatment, and they're concerned about what their future might hold. Now, an observational database is nothing more than a compilation of patient journeys. And sometimes we get caught up in the numbers and we start talking about the fact that we've got a 1,000,000-person data set or a 1,000,000,000-person data network that we're all collaborating on. But ultimately, we need to think about each and every individual that's in that data set and their journey and their experience. Because every patient's journey is quite different. You've got some patients that are going to have different disease course and different treatment choices and different outcomes. But ultimately, a database is nothing more than a compilation of these journeys, and we're trying to learn from those experiences. So what are the kinds of questions that a patient might care about if they were to be able to ask along their journey? Well, they might be interested in understanding, for other patients that have had that disease, what treatments did they choose? Or which patients chose which treatments? They might be interested in how many patients experienced an outcome after they've started a treatment. These are information that we can answer from these data. Patient might be interested in what's the probability that I'm going to be the one to develop this disease? Or the question of what's the probability that I'm going to be the one that experiences this outcome after I've taken treatment? We can answer these questions from the data. And we might be interested in understanding, well, does this treatment cause the outcome? Or does one treatment cause the outcome any more than some alternative treatment? Because if I'm making a decision, I might want to know about those effects. And we can answer these questions from the data. So as we've been classifying these types of questions, we've been putting them into these three buckets: clinical characterization, patient-level prediction, and population-level effect estimation. And what I want you to think about is clinical characterization is answering the question, what happened to them? To the patients who have already gone through their journey, what can we see that's happened to them so that we can count and tabulate and understand their experience? Patient-level prediction is going to answer the question, what will happen to me? So I want to infer from the experience of others the experience that I can anticipate for myself. And population-level effect estimation is going to answer the question, what are the causal effects? Not just that something's going to happen, but why it's going to happen. Now, we've been classifying these as three different types of evidence, but in fact, this information is quite complementary to inform the patient journey. If we are going to generate clinical characterization and understand what happened to them, that can be useful in its own right, but it can also be the precursor to start answering, if that's what's happened to them, now let's answer the question, what will happen to me, and answer a prediction question. Or we might be interested in understanding, now that we've identified causal effects through our population-level estimation, we can start to think, can we use those causal effects to actually predict what's going to happen to me? So we want to think about, while we've classified these as three different groups, and we're going to give you three different presentations, we want you to understand that this is all one big picture to advance how we can think about evidence to inform medical decisions for a given patient. We think all of this evidence is quite complementary. But one key distinction is that clinical characterization is nothing more than observation. I am simply tabulating what happened to them. No inference, no statistics. I'm just telling you, what did we see? Patient-level prediction is a question of inference. Given what we have seen, what can we project would happen? Population-level effect estimation is causal inference. It's not just being able to say what was going to happen, but it's also saying why is something happening. These are different types of statistical modeling problems that we need to handle slightly differently. We will contend that there's one framework that we can follow for all of these types of problems, but the specific solutions for each of these might be a little bit different. And that's what we're going to lay out for you today. So, generically, I just described the journey of a patient, but let's get more specific into a specific disease state. So let's think about a patient with depression. So what would that look like? A patient would have a diagnosis of major depressive disorder. They may get some treatment, like they will be exposed to an SSRI, and that patient might be concerned about whether or not they're going to experience some sort of an adverse effect, let's say like ischemic stroke. And of course, as we've said, that patient's data is going to contain a rich collection of other information that may or may not be useful. So we all have this kind of, at least I have this model in my head that this is kind of what the data looks like, more or less. Unfortunately, the reality of it is real data in a real database is a lot more complicated. What I'm showing you here is a screenshot of a patient profile from an administrative claims database of a patient who had depression, was exposed to an SSRI, and sometime later had an inpatient hospitalization for stroke. And you can see all of the data of all of their conditions and drugs and measurements and procedures and visits. This is a visualization to help you understand the data. If that's exciting to you, you need to go check out Siegfried Gold's poster presentation that's outside, where he goes through the tool that we've developed as an OHDSI community. Now, Anna stands up just about every single OMOP and OHDSI meeting and says, to do analysis, you've got to understand the data. And so, just to preempt it, I'll just say that, Anna, you're right. The whole point of this is that we've got to understand the data before we can actually go to answer questions. And we think that it's important for you to understand that this is what patients really look like. It's not just the simple cartoon that I drew up before. And unfortunately, it gets even more complicated because every patient's journey is different. Every patient's experience is quite different. We've got some patients who have a large number of comorbidities, many different concomitant medications. We have some patients who have extreme health service utilization in the data. All of these patients can answer the question, they've got depression, they took an SSRI, and they've potentially had a stroke. And yet, our challenge from an analytic perspective is how do we take the data from these journeys and turn this into evidence to help the next patient who's going down that journey to help their disease. So if we were to translate our generic questions into something more specific that could help a patient with depression, a patient with depression might be interested in, now that they've been diagnosed, which antidepressant treatment should I take? They might be interested in the past, which patients took each of the individual antidepressants. And they might be interested in, now that I'm going to be a patient who's going to take an antidepressant, what's the probability that other patients in the past have had an adverse event like a stroke? Really what I'd be interested in knowing is what's the chance that I'm going to develop depression in the first place? Or as it relates to that outcome, now that I know I've got depression, what's the probability that me, given my journey, given everything you know about me up to this moment in time, what's the likelihood that I am going to have a stroke? And of course, we want to answer the question, do SSRIs actually cause stroke? If we could understand the effects of a particular product, that'd be useful. But it's also important for us to complement that with answering the question, does an SSRI cause stroke more than some alternative treatment like an SNRI? These are all complementary questions that we think all of the evidence is needed to help a patient on their journey. So, what should a patient with major depressive, how should a patient with major depressive disorder be treated? We were just taking a look at the primary treatment guidelines published by the American Psychiatric Association for Major Depressive Disorder. Actually, just show of hands, how many folks here have done research in depression? Not very many. Good. So I can say just about anything and you won't know. That's helpful because I don't really know much about depression. But I read this document because I thought it'd be useful to understand, well, what is the guidelines that are being provided to other clinicians about what they should do? And here's what the guideline says about pharmacotherapy. It says, the effectiveness of antidepressant medications is generally comparable between and within medications, including SSRIs, SNRIs, TCAs, MOAs. Therefore, choose a medication based on the following. Patient preference. How does the patient know which one they want? The nature of prior response to medication. Well, I just got diagnosed, so I haven't taken any other drugs, so I don't know about that. The safety, tolerability, and anticipated side effects. Okay, so I might know about side effects that other people have experienced, but how do I know what's the chance that I'm going to experience the side effect? And the pharmacological properties, and then the cost. Okay. So I don't mean to belittle. This is the current state of the art, and this is what we're trying to do with depression. The question is, if this is actually true, if this is what we believe as a medical community, how could we provide evidence to supplement this so that people really can think about what should they prefer and why? What is the nature of their response going to look like? How can we understand the anticipated side effects? How is this going to be changed based on your patient's, your individual experience? So as George highlighted, we conducted this large-scale treatment pathway study last year, and it answered a very simple question. What do patients with the disease actually take? And we looked at major depressive disorder. We analyzed and said, so given that guideline, you'd kind of expect that patients would make a lot of different choices. What I'm showing you here in this picture is the results from across our network. This first circle is the first line therapy that patients take. The second circle is the second line therapy, third line, so on and so forth. And if you guys are getting like dizzy in the back because you're seeing all of this bright color, each color is a different drug. The fact that it's like this crazy rainbow is because lots of patients are making lots of different choices. There is no consistent standard by which patients with depression are being treated. We got 17% that are starting on citalopram, 16% starting on sertraline, 14% starting on escitalopram, another 8% starting on bupropion. We got some on fluoxetine, so on and so forth. Because the guideline basically says make a choice, patients are making choices, lots of different choices. The challenge is, if that's what's actually happening, how do we learn from it? So, importantly, within the OHDSI community, we can start to get to the bottom of this type of question. These are the data partners who have participated in that initial study that George outlined. And as George kind of humbly stated, this was a study that we thought, this will be kind of fun to do. And in a few weeks, we had gone from idea in the bar in this very building to a protocol, to code executing around the world across all of these sites to be able to start to answer the question, how do patients actually get treated? The protocol, you can take a review. Importantly here, we started with a data network of over 250 million lives contributing to this study. But after we applied extremely strict criteria requiring 4 years of observation and 3 years of continuous therapy, we were left with 264,000 patients. Best I can tell, that's probably the biggest study that's ever been done for this length of time, and it would have been impossible if any of us tried to do it ourselves with our own individual data source. No one data source is going to be answering this question. And probably most importantly is that patients with major depressive disorder do not look the same everywhere in the world. What I'm showing you is another screenshot from the paper. This is the results from Korea and the US and the UK and Japan, from EHR systems, inpatient and outpatient, and claim systems from Medicaid and Medicare and privately insured populations. And the one thing that you can see across all of these pictures, if they look like crazy rainbows to you, it's because everywhere it's a crazy rainbow, and yet that crazy rainbow is different in every single site. We see differences by data source, we see differences by health systems, we see differences by geography, we saw differences over time. The only constant was variance. One of the things that we found actually was that 11% of the people in this data set of 250 million patients and 260,000 patients, 11% of the depressed patients had a patient journey that was all to their own. That is, that patient took a sequence of treatments that nobody else across the entire network had that same exact sequence of treatments. So just think about that. 1 in 10 patients' journey. When you ask the question, patients like me, go find me the patients like me, there was nobody like them for 10% of the patients. Right? If you think this is cool and you'd like to do characterization, check out John's demo. He's actually, in collaboration with several folks, has developed a tool so that we can explore not just the diseases we did in this paper, but actually explore any disease you're interested in, because we think this has broad utility and value to just characterize populations at mass. Okay, so we want to be able to answer the question, which patients chose which antidepressant therapy? So if I go back to our cartoon, I'd like to be able to start with the treatment and look backwards in that baseline time. And to do this, if I want to be able to understand which patients took which treatments, I need to be able to create a cohort for all possible antidepressant treatments. I need to be able to summarize all of their baseline characteristics, and then I need to be able to systematically explore those differences. So the only way we're going to be able to achieve that is if we agree to a standard way to construct cohort definitions that can be consistently applied across different disease states, across different treatments, and across different databases. So within the OHDSI community, we have developed a tool to standardize cohort construction, and Chris Knoll will be doing a demo of that tool out to showcase. But using this tool, we're able to create standardized cohort definitions for every antidepressant treatment. So we have 17 that we looked at based on the prevalence across different databases. We looked at all of the different drug treatments, whether they be SSRIs, NRIs, bupropion, TCAs, etc. We also can think about interventions that are not drugs. So we looked at psychotherapy and electroconvulsive therapy as alternative interventions that a patient with depression could have. And by having standardized definitions, we can run that across multiple different databases simultaneously. I'm just giving you a snapshot here of three different populations. We've got a privately insured claims database, a Medicaid population, and a Medicare supplemental population. The point here is to say consistent definition across different databases start to provide us the scale to start asking questions that we probably haven't been able to do in the past. So we got 17 different depression treatments. So now that I've got 17 cohorts, what am I going to do with them? What I'd like to do is just summarize who are the patients. What can we learn about them prior to their treatment? And so I'd like to be able to summarize their demographics. I want to know their age and gender. I want to summarize their conditions. So what diseases have they had in their history prior to exposure? I want to know what other treatments they've taken. I'd like to know what procedures have been administered, what their laboratory response is. I'd like to just know the overall welfare of these patients, whether that be risk scores or just density of data that we have about a patient. So importantly, this is not different than what we all do right now when we conduct an observational study. We all publish papers that have a table 1. And in table 1, you usually take your one cohort, you pick your 10 or 15 variables that you think are important, and you enumerate them. You tell me what is the population I'm interested in. Not doing anything different, just going to scale this to be just a little bit bigger. So how much bigger? Well, we've got 17 treatments, that's 17 cohorts. And because I don't know the 15 or 20 things that I might want to cherry pick to put in my table 1 of my paper, I'm just going to summarize everything. Between all of the drugs and all the conditions and all the procedures and all the measurements, we can have about 230,000 baseline covariates. I can run this across every database. We've just done 4 for illustration purposes. So that's going to give me 15,812,000 summary statistics. Now that's big. We've actually created big data out of big data. We've now got millions of summary statistics. I'm sure it's going to happen again this time around, so it's also preempting it out of the table. We're not data mining. Nothing I'm describing here is data mining. What we're doing is we're applying a systematic process consistently across questions that matter. This is large-scale analysis, but every one of those summary statistics is absolutely no different than the number you would put in a table 1 of your paper. It's just that we're going to generate the table 1 for all your papers. And we're going to not allow you to not going to tell you which 15 or 20 variables you're going to cherry pick. We're going to provide you all the information that anybody might want to look at because they might be interested. So, just to cherry pick, I could think about one individual variable. So let's just think about health service utilization. How often did a patient on an SSRI go to the doctor in the prior 1 year? And what I'm showing you here on this graph is on the x-axis, I'm showing you the mean number of visits in the last 1 year. On the y-axis, I'm showing you all of those different antidepressant treatments. In these various columns, I'm just showing you 3 of the databases. So I'm showing you 3 populations. And what I want to point out is, remember that clinical guideline that said all of the treatments are more or less comparable? That might be true in terms of the effectiveness of the treatments, but what we're showing you right here is even though the treatments might be comparable, the patients who are getting the treatments are quite not comparable. What you can see is that some things like psychotherapy, patients had a lot fewer prior visits. Electroconvulsive therapy, they had a lot more visits. Even across drugs in the same class or drugs across classes had wild differences in their health service utilization in the prior 1 year. So even though these are treatments that we think, oh, they're the same, or maybe I'll just create a drug class of all SSRIs because it's all the same thing, actually, the experience of individual drugs and the patients who take them is very different. And to make it even worse, if we look across different databases, we see that the types of differences you observe are not consistent. It's not the case that you can say, well, sertraline's got more prior utilization everywhere. It might be the fact that sertraline has more utilization in one population and less prior utilization in another. You'll only know that if you systematically explore and summarize the data. So just pivoting, so I just described how we can think about baseline characterization. At scale, generate the evidence so that we can understand what's going on with patients before they ever take treatment. But of course, the patient also wants to know what's happened to those other patients in terms of outcomes that have been experienced. So how does a patient go about finding current evidence about outcomes? Well, they could read guidelines, because I know every patient just loves to read clinical guidelines. If you did read the American Psychiatric Association treatment guideline, you'd see that this big long table of listing the side effects. And basically what it does is it lists out there's individual side effects, It says what drugs are associated with the effect, and it even does us a solid and provides what treatment should you give if the side effect is observed for the patient. And so you can see that various antidepressants are associated with falls and GI bleeding and hepatotoxicity and insomnia and nausea and vomiting, and of course I just couldn't even go on further. We could also look in the published literature. We could see whether or not there's any studies that implicate the effect, and here we looked at a published book called Drug-Induced Diseases, which lists out a whole bunch of side effects we might be worried about, and we can see information about which drugs are purported to be associated. And we can see many of the antidepressants are associated in this book, according to these authors, they're associated with hepatic issues. Of course, the incidence of those hepatic issues is not known. There's been some case reports published, maybe there's something in spontaneous data, but there's no incidence to tell us how often that's actually happening. Of course, we can go to our tried and true product label. John talked about this last year. We could read the product label and look for information. And if it's an important side effect, it might be in a black box, or it might be somewhere else in the warnings and precautions, or we could go to our laundry list in the adverse reaction section and hope to find information about what outcomes might matter for patients. So how does observational data contribute to this? Well, we can look in the literature and we can see if there are observational studies that have been published on any particular issue. So here I'm interested in SSRIs and whether or not they cause stroke. And actually we were in luck to find just a couple years ago there was a systematic review of observational studies published in the Journal of Neurology. And it had this forest plot that showed for ischemic stroke, lots of different researchers used different methods on different databases and got different conclusions, just like what George showed you this morning. So we had some people that did nested case control studies and said there was really no effect, but there was some uncertainty. We had some people who showed an effect. We had some people who actually showed a large effect. Overall, this I-square, that's a measure of heterogeneity. If anybody's a meta-analysis wonk, usually the rule is if the I-square is greater than 0.75, don't think about doing a meta-analysis and summarizing together. So you can see what they've done here is done a meta-analysis and summarized together to come up with the conclusion that the relative risk is 1.48 with a confidence interval from 1.08 to 2.02. So if I'm a patient, I might have to read the Bach study, the Chen study, the Tifero study, the Smoller study, the Pan study, the Hung study, and be a little bit confused by that. And I could read this meta-analysis and maybe get some resolution, right? Because this is the summary of all the information. Well, the summary from this meta-analysis was, since there's heterogeneity among studies and possible confounding from depression that could not be fully excluded, further well-designed studies are needed to confirm this association. So after we've done all of these studies and we do a meta-analysis, our conclusion is let's do more studies. The challenge is, as George outlined, that if we think about the world as a bunch of dots in a space and we don't understand the entire surface, we will continue to get to this kind of confusing state where we're at. So this question is one of effect estimation. Martijn will get into how to solve this problem. What I want to do is just step back and say, well, what problems should we try to solve? And what I'm interested in is the question, how many patients experienced outcomes after treatment? So what I want to do is look forward after they've been exposed to the outcome. I want to create not just all of the cohorts for all the treatments, but I want to create a cohort for all of the outcomes that I might be interested in studying. And if I could do that, then I could summarize the incidence to know how often every outcome happens for every treatment. And I might be interested, if I could do that, I might be interested in asking, well, what subpopulations am I interested in studying? Because, of course, me as an individual, I belong to some subgroup that might be of interest to me. If you guys are interested in incidence rates and how that could be done in a systematic way, there's several posters in our clinical application section, including one by Urmila and one by the team at Azu University, talking about different disease states and how you can estimate incidence rates. So definitely should check that out. So how are we going to do this? Again, it comes back to standardizing our cohort construction. We need to be able to create a consistent way to define phenotypes so that we can look for outcomes just in the exact same way that we need to be able to standardize the way that we would define exposures. So how is that done currently in practice? Well, there was actually a recent publication that looked at SSRIs and ischemic stroke. And what they published is they said, well, we looked for outcomes by finding patients who had a first hospitalization of ischemic stroke as defined by ICD-9 433, 434, 436, And it had been previously validated, so we had a good positive predictive value. And we wanted to make it more accurate, so we also required that a patient have a CT or an MRI during their hospitalization. Really good study, really good research group, well-intentioned, doing the best they can, just like George outlined. Okay. Just last year, there was a terrific paper that was published by the FDA looking at ischemic stroke, specifically for the anticoagulants. I don't know, actually, is David here? Is David Graham here? No? Okay, well, when the FDA goes back, tell him that his paper's awesome, it rocks. We're using it a lot for our symposium because it's actually a really good demonstration of how a paper could be done. In that paper, it actually gets more explicit and provides you how you could define ischemic stroke. And in the Graham supplement, it tells you that ischemic stroke is defined by 4 through 3.x1, 4 through 3.x, exclude subcode x0, 4 through 6. It has to be in the primary position of an inpatient hospitalization record. So these are different definitions. I can't tell you which one's right, which one's wrong, but I can tell you that they're different, and that difference could be possibly part of the explanation for why one might get different answers if we're trying to explore that surface that George laid out. So what we need to know is we need to be able to know the trade-off of the sensitivity and specificity and how that would impact our ability to generate reliable evidence. We don't have a solution to this problem, but we recognize it's a key area that we'd like to encourage all of you to think about. The things that we think are steps in the right direction is that we need to create standardized definitions, and we think that that large-scale characterization of baseline characters can help a lot. If I can see who are the patients who have journeyed to a particular disease, I might be able to see, are those the patients I really want? We can review patient profiles. I showed you a couple examples of that, and I'd encourage you to check out the demo, because you can actually learn a lot just by looking at the data, as Anna keeps imploring us to do. We need to be able to think about comparing alternative definitions. Rather than us choosing one and assuming it's right, if there is uncertainty, explore the uncertainty and embrace the fact that you might get different answers, because that uncertainty is actually knowledge. And of course, if you're not sure about how to create a rule, maybe you shouldn't create a rule at all. Nigam Shah and colleagues have talked about how we can actually do predictive models for phenotyping, and encourage you to check out the work that he and John and Chris are doing in the cohort definition work group. So, taking the best we could, reviewing the literature, reviewing alternative definitions, exploring patient profiles, and looking at definitions, we have scaled many different outcomes known to be associated with antidepressants. 22 outcomes here. So these were the things we found from the literature, from the guidelines, from the product labels, things that might be associated with antidepressants. And there's the list, everything from rare serious side effects to common nuisance effects. So the point is that if I want to calculate an incidence rate, I want to do that at scale. So I've got 17 treatments, I've got 22 outcomes. I might want to stratify in various factors like age and gender and maybe prior depression or prior diabetes. I've got 4 different databases, so that means I'm calculating 8,976 incidence rates. I'm not doing data mining. This is large-scale analysis. Every one of those incidence rates is just like you would calculate if you were going to write an individual paper about one incidence rate. You create a proper cohort definition, a proper outcome definition, you think about your time at risk, you calculate a number, and you say that this is the characterization of what's happened to them. We can scale that by using common standardized tools so that if the problem you're interested in is you are interested in sertraline and ischemic stroke, but I'm interested in bupropion and diarrhea, we should be able to follow the same scientific process to generate the same quality of evidence, no matter what that number might be. So, what is the incidence of ischemic stroke for patients with an SSRI? We, in fact, have a new tool that's just been released in Atlas that'll actually show you the incidence for every possible treatment and every possible outcome across every possible subgroup. And encourage you to check that out. In the interest of, let's see. Actually, I'm going to do it. What the hell? All right. Let's try this. All right. So, I apologize for those that sat in the back who are going to have a hard time squinting. Just trust me with whatever I say, I guess. So, I'm in Atlas. Atlas is the common analytics platform that the OHDSI community has been actively developed on, providing features to do characterization, effect estimation, and patient level prediction. I'm just talking about characterization. But what I'd like to be able to do is think about all of the possible target cohorts I'm interested in. And we can see we've defined all of the different individual treatments, all of those different antidepressants, including just replicating exactly what has been in prior studies. We can also define all of the possible outcomes that we're interested in. So I'm going to define ischemic stroke, both the way that David Graham wants to do it, as well as the way that the Lee and colleagues want to do it, as well as all of these other outcome definitions. Once I've defined my target populations, my incidence rates, I can think about defining my time at risk. Once I've defined my time at risk, I can think about defining stratification factors, ways that I want to stratify the population to understand whether or not the incidence rate is consistent or similar. It could be any collection of things, like here I've got age and gender, type 2 diabetes, whether they've got prior heart failure. Specifically here I'm thinking about ischemic stroke as an outcome. Those are risk factors known to be associated with stroke, so maybe I want to understand whether or not I'm in that group. We can run an analysis across any set of data sets. I've just done here a few just to illustrate it. And what I can see here, I don't even know what I've picked on here. So I'm going to look here for SSRI new users and the outcome of ischemic stroke. And what I can see is in the Optum data set, I have 700,000 patients who are new users of SSRIs. 985 of them experienced an ischemic stroke during my time at risk, which was the period of exposure. That gives me an incidence proportion of 1.39 per thousand persons, an incidence rate of 2.62 per thousand persons. We can see how it compares to other populations like Medicaid and the Medicare supplemental population. Medicare supplemental population, an older population, we can see that they have a much higher incidence proportion. Now that's all fine and good. I've got incidence rates. I could change that to any outcome I'm interested in. Maybe I'm interested in switching that to myocardial infarction. I can explore the same kind of characterization of how big the population is, how many had the outcomes during the time at risk, and what is that resulting incidence rate. Okay. But I might also want to be able to explore to say within a particular subpopulation, what is the effect of all of those stratification factors? So I knew already that the incidence proportion was 1.5 per thousand person years, but what is it for people who are men? It's 1.8. And how does it break out by age? Well, I can see in people that are young, it's pretty low. In people that are older, the incidence of stroke is much higher. And what about people with diabetes? Well, it's about 4.5. And what about people with heart failure? It's actually almost 11 per thousand persons. This tree map, that I'm going to struggle to get over to, is breaking out our entire population of 700,000 patients who are new users of SSRI, breaking it into all the possible ways that a patient could be divvied up across all of these different stratification factors. And it's colored right now based on the incidence rate. And what I want to call you to is blue is a low rate, red is a high rate. And it goes in this gradient here. What I want you to notice is if you're thinking about ischemic stroke for this big population of SSRI new users, what you can see is the vast majority of patients actually have a remarkably low risk of ischemic stroke. If I'm one of those patients and I'm in one of those low groups, I might be kind of interested in thinking, well, that's what's happened to them. That might be useful. And as Peter's going to go through the patient level prediction, he might be able to refine that to tell me what's going to happen to me. What we can see is that there's a small population way up here. It's only 1,000 patients. That 1,000 patients represents less than 0.2% of the population. But in that 1,000 patients, those are people who are male, over 75, have type 2 diabetes, and have heart failure. That whole set of data in that patient journey. And for that small subset, the incidence rate is 38 per 1,000 person years. Okay? So small subset of people have a very high risk. Large sets of people have very low risk. By being able to systematically explore this kind of information, we're getting closer to understanding what has happened to them and splitting up the population to understand where you might fall in that continuum. Now here's the thing, I can only tell you what has happened to them, what has happened in the past to the population I had, and we need to be able to look across multiple different populations because different populations have different characteristics. What Peter's going to talk to you about is how do we translate some of this piece of information of what happened to them and model that to be able to start to answer the inferential question, what will happen to me? Thank you. They're excited, Peter, they're excited. They can't wait. All right, so the point here is that this is as good of an incidence rate as anybody can calculate. If I were to publish a paper just on an incidence rate, I would calculate it this exact same way. This is not data mining. I'm not doing any data drudging. What I'm doing is saying there's a systematic way that we can generate insights from data to look consistently and reproducibly across every database, across every population we care about, for every outcome we care about. And if we are able to agree to common standards and best practices, we now have the ability to generate the evidence at scale that we can all take advantage of as a community. So what is the journey towards reliable evidence? What you're going to hear from me and Peter and Martijn is that we really think that there's three fundamental components we need to focus on. We need to think about evidence generation. How do we produce evidence from the data? How do we take those journeys of the individual and turn that into summaries that tell us something that's insightful? You can't do generation all by itself. You need to think about evaluation, because it's not just about producing a summary statistic, it's about knowing that that summary statistic you produce is valid, reliable, and meaningful. So how do you know the evidence is reliable? That's one of the challenges we want to think about. And then finally, we really need to, as a community, think about evidence dissemination. How do we share evidence to actually inform decision making? From the perspective of clinical characterization, I'm going to leave you with these insights. From an evidence generation perspective, I'd argue that our path forward needs to be to follow a standardized process. We can and should use open source code that promotes transparency, and use validated software so that we all have the same level of confidence that we're doing the right analysis consistently every time. But analyses that we develop need to be scalable. If you can do a study well for one question, we as a community can do that same study well for all questions. And so we want to have analysis approaches that are scalable to many exposures, many outcomes. This is not data mining. This is a large-scale thinking about how we generate evidence. And if we're going to do this, we need to be able to replicate across databases. We can't be a single dot on the surface that George laid out and just a hope that our answer is consistent across the world, across geographies, across health systems. We need to actually answer that question because patients deserve it. From an evidence evaluation perspective, I would argue that we need to apply tools to explore the patient journeys and the population characteristics to assess the validity of these cohort definitions. It should not be acceptable that you say, hey, I referenced this paper from 10 years ago in another geography in a different coding system, but it said there was a good positive predictive value, therefore I'm just going to assume my definition is good. We can do better than that as a community, and I'd argue that it's really incumbent upon us to do that. One possible path forward is to make these comparisons across populations to really examine the study heterogeneity. This is an area that I would make a plea for those who are saying, hey, I'd love to get involved in Odyssey, what could I do? We need to solve this problem of phenotype validity and evaluation. And if anybody's interested, I know Nigam and John and others would be delighted to talk to you about that, as well as myself. Finally, last but not least, evidence dissemination. I would argue that if we're going to do large-scale characterization, we need to get away from thinking about static reporting. We shouldn't be thinking the end game is a PDF document published on an 8.5 by 11 piece of black and white paper in some journal that's behind a firewall. We need to actually think about how do we get the evidence in the hands of patients that can actually make a real decision. And if we're going to generate the evidence at scale, I can't do this one number at a time, one question at a time. I need to actually have an exploratory framework where I can explore all the information as much as I want it. And I would argue that what I've just described for you, while I think is valuable in its own right, I would argue that it's a required supplement to which is the real meat of our meeting, which is what Peter and Martijn are going to describe to you. That characterization provides the context of what will happen to them, but where we really want to go is what's going to happen to me and what are the causal effects. And this provides that foundation to help you interpret those analyses. So with that, I want to stop. I want to encourage you all to join the journey. And we're going to take a 15-minute break. And when we come back together, Peter is going to take the next step on this journey, which is to describe how we can think about doing patient-level prediction.